Hey folks, welcome to 8.6, Osmosis and Dialysis. It's the last section of chapter 8. So we are going to wrap up this chapter today. We've covered a lot of things here. Types of mixtures. We have solutions, colloids, suspensions, and we decided to focus on solutions with small molecules. Talked about how we can dissolve covalent ionic compounds in a solvent. We talked about how we can quantify the amount of solute with concentration and all the different units of concentration we can have. We used concentration to determine all sorts of dosage calculations in medicine, and including flow rates for IVs. We talked about dilution, and now we're going to talk about osmosis and dialysis. So let's get to it. So a solution is a homogeneous mixture, right? Why is that? Well, what does that mean? Well, so first off, remember that homogeneous means evenly distributed throughout. Okay, and there's some time component to this, right? So I really like this picture here. Imagine we just have a beaker of water, and we're gonna put like a drop of dye or food coloring in it. Okay, and that food coloring obviously drops on the surface in a particular spot, and it'll fall in and kind of start to spread out. And that act of spreading out is important. So spreading out is diffusion, right? And it's going to have to do with the solute particles just making their way, pushing their way through and around all of the solvent, right? So first it'll spread a little bit and then a little bit more, and then eventually, right, it'll be evenly distributed throughout the entire solution. So this process of evenly distributing is called diffusion. And here's the quote, right, that we can say about this. Um, Remember that solute and solvent molecules are both there, and they are constantly in random motion. They're constantly in random motion as a result of the kinetic energy that they possess. And the higher the kinetic energy, the more motion, right? Kinetic energy is the energy of motion, right? And so if they're a higher temperature, that's a higher average kinetic energy. That's faster moving molecules. And you know from experience that a warm solution will diffuse or mix faster than a cold one, right? And that's because there's more random motion. So the random movement of the solute particles through the solvent particles is known as diffusion. Now, another way to think about this is in terms of concentrations, right? And here, when you first put the drop of dye in, right, it's highly concentrated, all in one spot, and all around it, there's no dye molecules, right? The concentration is essentially zero around here, and so there's going to be diffusion from area of high concentration to the area of low concentration. It just starts to spread out until the concentration is equal everywhere. Equal concentration everywhere is also known as evenly distributed, right? So the net result of diffusion is the movement of particles from an area of high concentration to an area of low concentration until the two areas have equal concentration. So this sounds kind of like an equilibrium because it is, okay? So that's like the basic idea of how solutions end up homogeneous is through diffusion. And uh, diffusion occurs, as we said here, until the two areas have equal concentration, unless there is something in place to prevent diffusion from happening or occurring naturally. And that's where we're going to go next. So diffusion can be controlled by a selectively permeable membrane. One example of this type of membrane would be a cell wall or cell membranes, right? It can control diffusion and allow only certain substances to pass. So if there were no such thing as if cell didn't have a selectively permeable membrane, like the picture here in the top right, then the concentration of everything inside would be the same as the concentration of everything outside, right? Then they would diffuse in and out until there was equal concentration. Okay, but that's not the case for a cell, as you probably know from biology and otherwise. Um, we have what's called a concentration gradient, that there's a difference in some of the concentrations. So down here in the bottom left is an example of cell membrane controlling what enters and leaves the cell. So it's a selectively permeable. So some things can get through and some can't, and it controls diffusion. So clearly, here in the bottom left, right, these, whatever the blue dot may be, there's a much lower concentration inside the cell than there is outside the cell. But all those outside are not allowed to diffuse in. So this is quite an important phenomenon, and it allows the cell and many other things to maintain highly regulated concentrations of certain things. And once again, this term concentration gradient is if two solutions of different concentrations are separated by a selectively permeable membrane, it creates a concentration gradient where there's just different concentrations, right? So the kinds of things that can move through a cell membrane depend on the size and the charge. Okay, so here is a chart showing at the top things that pass through more easily, so they're highly permeable. Small non-polar molecules like oxygen, carbon dioxide, and nitrogen are highly permeable. Then small polar molecules like H2O, it's small, it's still uncharged, but it's polar. And then less so would be large uncharged polar molecules, right? They're like kind of turned back, but some will get through. Ions are even less permeable because of the charge. And then large charged macromolecules, proteins, polysaccharides, triglycerides, fats, will not get through very well. They're low permeability. So the most important factors in determining whether a solute can go through a cell membrane are size and polarity, right? Or size and charge. If we look at table 8.6, we can see the concentration gradients that occur inside and outside the cell. And notice they are all different, but also very, very different in each row, right? So sodium ions inside the cell have a concentration that's best at 10 millimolar, right? 10 millimoles per liter. But outside the cell would easily be 140 millimolar. So a huge concentration gradient, as well as for chloride, 4 inside the cell and 100 outside the cell. But it's not always smaller inside the cell. For example, potassium ions have a higher concentration inside than out. So the permeability goes both ways. And calcium ions have a huge gradient at 1 times 10 to the minus 4 inside and 2.5 outside. So almost 10,000 times difference. So the cell membrane is really quite good at maintaining this concentration gradient inside and outside the cell. Here's a question about permeability. So select the correct statement regarding solutes crossing a membrane. And pause it for yourself. And we find that B is the correct answer, right? Water molecules, small, uncharged, are more permeable than ions. Yes, that's true. They're higher up on the chart. Okay, all the rest of these are false. Okay, let's stop here and we'll come back for part two, and we'll start with what kinds of things can move or diffuse across a cell membrane to change concentrations. So we'll come back for that and continue in part two.